So we're starting on our newest unit, which is about reproduction. And when we first talk about reproduction, we do have to remember that there are two different types. There is asexual reproduction, which a lot of times we can forget about, and also there's sexual reproduction, which is what we're used to thinking of as producing more organisms. But when we think about reproduction, what is reproduction? What are we making? And when you see the word reproduction, you should remind yourselves that this is to make more. But what are we making more of? A lot of times that means that we're going to be making more cells or more organisms. So cells or organisms. When we're making more cells, usually there's going to be two reasons why we would need to make more cells. That could be simply to grow or to repair. If we're making more organisms, usually this means that we're going to try to continue the species, which for a lot of animals or a lot of organisms in the world, this is kind of all they do. The only goal they have in life, the only purpose they have, is to try to continue their species. There's a type of fly that literally grows out of the water, and as it grows out of the water, it has about 48 hours to live and reproduce, if it doesn't get eaten by something else. But, so reproduction, we can make more cells. We can make more cells so that we can grow. We can make more cells so we can repair. Or we can make more organisms, complete entire organisms, and this is going to help us continue the species. When we talk specifically about asexual reproduction is usually a form of growth and repair. It's mainly to grow or repair. But we can make more offspring. The interesting thing is when you reproduce asexually, the offspring are going to be genetically identical to the parent. And a lot of times we'll refer to this as a clone of the parent. Okay? A benefit or an advantage of asexual reproduction is that this is a quicker process because you don't need to find a mate. So over on the right hand side of your notes, you do have some examples of asexual reproduction. The first, where you see in the strawberry plant, the onion bulb, and the potato plant, these are actually examples of what's known as vegetative propagation. Generally, what happens in vegetative propagation is that you have a main parent plant, an original parent plant. The parent plant is going to produce what's known as a runner. Here's the runner. And the runner is actually a stem that will grow out and produce an entirely new organism. So what I want you to do is actually to take a minute and click on the link that is attached to this picture. In the link in the picture, you're going to watch a video about regeneration. And regeneration is a second type or another type of asexual reproduction. And in this type, you'll actually see an example with sea stars. It's a really quick video, but I do want you to take a minute to do that and write down two key points or two notes on the sea star. Then, if we look over here to the right, we have two more examples of asexual reproduction. So let's kind of number these just to keep these in mind. We have one example, which is vegetative propagation. We have another example, which is regeneration. We have a third example known as budding. And in budding, this little guy here actually pops off and becomes like a mini-me version of the bigger polyp. This organism is known as a hydra, and this is actually an underwater animal. kind of similar to a jellyfish. And notice that it actually has two polyps forming, and these eventually will fall off and become a whole separate organism. And they are all genetically identical to this original parent organism. Then when you look over here, this is your fourth example, which is binary fission. And in binary fission, you divide equally into two. And this is really what we see happening in most of asexual reproduction, and this is what you see happening in mitosis. Okay, so an easy example of that. And then a final example, and I don't have a picture of this for you guys. Another example would be sporulation. And sporulation will happen in fungi, where little spores are produced, that little dusty powder if you kick over a mushroom. Are actually going to be able to form a brand new identical organism. So here are 5 different examples of asexual reproduction, but why would we want to asexually divide? Well, one of the main reasons for this is that you're going to want identical copies. And having identical copies of a cell is going to be important for growth in multicellular organisms, and repair. So imagine your skin cells. Your skin cells need to be exactly the same. They need to have the same genes. They need to have the same genes activated. And so another reason in this idea of growth, is that we need small units that function efficiently. So this is the idea of surface area versus volume. Okay, a very large cell is not going to be as efficient as a smaller cell. And if you think about the process of osmosis and diffusion, well, this is when we can have this problem, or have this be more prevalent. So with the idea of surface area versus volume, the higher the surface area you have and the lower volume you have is going to be the ideal sized cell. But then when we look at asexual reproduction, in asexual reproduction we're making identical copies of cells. But what causes cells to differentiate? Or what is differentiation? So if you look in this picture over here, you have an adult skin stem cell, and then you have these daughter cells, or daughter stem cells, that are growing from that. You want to take a minute and read over this little caption, this little image about adult stem cells, and just think about, well, why is the stem cell so important? Stem cells are really important when we talk about differentiation, okay? Because many cells will perform specialized functions that are going to be based on their genes that are activated and their shape. So that idea of form versus function, or form and function. Now, this bottom box here, I do want you guys to take a little time after the notes to find an example in the human body of cell regeneration. And you want to be careful about your source that you're getting this from, and you don't want to choose a skin cell. Okay, but moving on to the last portion of our notes, we do need to talk about, well, what needs to be done before a cell can divide. Okay? And so before a cell can divide, and when we say divide, we actually mean multiply or make more cells. so you don't want to get that confused. But when a cell is going to divide or make more cells, we are talking specifically about asexual reproduction. And in asexual reproduction, in order to make more cells or divide our cells, we call this process mitosis. In mitosis, we will actually start by replicating or copying our DNA. And the reason why we want to copy our DNA is to prevent the loss of DNA. Okay? If we're making two cells from one cell, that first cell that has the original DNA, well, we don't want to have to split that DNA in half and lose cells in our new daughter cells. We don't want to not have all of the information that we need. So, how does the DNA divide? We will be talking specifically about mitosis in the next section of notes, but we want to know a little bit about the DNA before we can move on. As it turns out, DNA is negatively charged. And this is going to be because of the phosphate, the PO4 group on the DNA. Now, DNA is actually going to, the double helix is actually going to coil around proteins known as histones to form a chromosome. So in this image, what you're seeing is this is the DNA. These little red circles are going to be the histones, and the DNA is wrapped around it. Now, what's going to happen is that our chromosome is copied or replicated, and this becomes what's known as a doubled chromosome. Okay. So here they're showing you this is a doubled chromosome. And one side of that is going to be one copy, and here's the other copy. Doubled chromosome. And I'm going to shorten it CHRMS. When you're looking at a chromosome, you do want to know that some shape like this, and I didn't do a great job right there, this is known as a single chromosome. And after we copy it, or after it's copied or replicated or duplicated, you're going to have a structure that looks kind of like this. Yeah, not my best job there. And this is now going to be known as your doubled chromosome. Okay. Each side of this chromosome, each individual side, is going to be identical. And these identical sides are known as sister chromatids. The other thing that's important here is that center structure that you're seeing is known as a centromere. And the centromere holds the two sides together, the two identical copies. And this is a region of protein to attach DNA copies together. And it also turns out that when we go through mitosis, the centromere is where spindle fibers attach and where spindle fibers can actually move the doubled chromosomes around the cell. Okay, and that's going to be all for today.